Hello students, welcome to EPG Pathshala. I am Dr. Ashish from Institute of Nanoscience and Technology, Mohali, Punjab. Today we are going to discuss a module which is nanostructures or design of nanostructure from hybrid system under the paper Nanobiotechnology. So after completing this module, you should be able to understand different hybrid structures. So we would actually discuss in this module different hybrid structure like polymer DNA hybrid structure. We would also know about different metal nanoparticles or nano containers, and how we can make these inorganic organic hybrid materials and how we can use for different applications so we would get to know from this module. Now we will discuss about other nanomaterial which are already we know in great details. So they can be this inorganic or carbon based nanomaterial. So we know about this metal nanoparticle like gold or silver based nanoparticle. So they are very they're interesting material because people have looked at last 20-30 years how to control their shape and size and how their property they completely changes based on shape and size. People are looking at also the quantum dot made from carbon based material. So there are a number of things which inorganic and carbon based nanomaterial has to offer in terms of the property. So they can be used for sensing, they can be used for therapeutic applications. So a number of things which are there, but I think at one point we have to also understand that many of these metal nanoparticle they can be also toxic. So there is a great deal of interest for making hybrid nanostructures or nanoparticle where you can have both inorganic or organic hybrid, where you refrain from using toxic metals. So when we talk about engineering the structure of this kind of hybrid material, one of the first example let's discuss is DNA block copolymer hybrid material. It can be called molecular chimera, and you can see here what happens is that here it's a DNA, single stranded DNA, one block, and another block is polypropylene oxide. Now polypropylene oxide is more hydrophobic, so it can form this kind of micelles where in water where all the DNA single strands they are exposed outside, and polypropylene oxide, that's the green color one, they are buried inside. Now this single stranded DNA that has oligodeoxynucleotide which is of 22 mer, and it has a molecular weight of around 6670. And if you look at the sequence of 5' to 3', now if you take the red DNA strand, which is just the complementary DNA strand of the blue one, then it can actually form DNA double strand. And what we see, if you look at the AFM image for A and B, A stand for the before hybridization with this complementary strand, and with the B, more or less the structure they spherical in nature, and there is not much of difference in terms of height. It only goes from 5.2 nanometer to 5.8 nanometer on the hybridization. They stay in spherical micelle form. So interestingly, we see when we take a longer template, the red strand which we take, which is it's much longer as comparison to the blue strand which is 22 mer, we take either 110 88 mer, and it has different segments like T, the strand with 110 mer, it has 5 segments which are complementary to the actual single stranded blue color single stranded DNA. And in fact what we see is the following. We see no more spherical aggregate, we rather see a rod like micelles. So that comes because the red, using the red strand as template, this blue strand actually forms this complementary hydrogen bonding at 5 different spaces, T110. It's also expected that T88 it forms in 4 places. So if you look at the AFM, you can see the length of that particular SM rod like micelles, which actually is something like 29 nanometer, but in the case of T88, the length of the rod like micelle is less. And look at this height image. That is, height is something like 1.9 nanometer, and you can also look at the spacing between two parallel strand, and that you can figure it out. The spacing between two parallel strand is 3.4 nanometer. So this is a very elegant example that you depending on the length of the complementary strand, you can either stay in spherical micelle, or you can completely change the structure into rod like micelles. So now we look at one example where DNA block copolymer can be used as drug delivery application in cancer cells. So in this, you can see in the top the DNA single-stranded block copolymer. It forms micelles with the hydrophobic polymer unit inside. Now then it actually can be hybridized with the complementary strand to form double-stranded corona. Now, in order to increase the efficacy of targeting to the cancer cell, foliate is known as a good receptor. So foliate can be also conjugated with this complementary chain, which is given by the red dot, foliate. Now doxorubicin, which is given represented by the green dot, that can be incorporated within this hydrophobic core, so it can actually go inside the core, doxorubicin drugs. Now the number of this foliate receptor can be varied, and in this example they have varied the foliate receptor, and they have preferentially put the foliate receptor in different parts of the micelles. Now when they have looked into the uptake of this DNA block copolymer micelles into cancer cells, this is CACO2 cells, they have seen when they have 28 foliate receptor, they are outside corona area more, then it has a good uptake, and as compared to when they are having only 11 foliate moiety outside, or even when you have 2 or foliate moiety outside, or even if you have put 28 foliate receptor, but there you have put towards the core of the micelle, you don't get good uptake. So it is important that this foliate receptor should be the outside of the corona of the micelles. In this example, Andreas Hermann group, they have shown, depending on how you conjugate your foliate receptor, that has an effect in the cell viability of the cancer cells. So first example, if you attach covalently, here all the foliate receptors are attached outside within the corona. You see the red dot. You can see here when you have, let's say, the red dot sitting outside and they are covalently conjugated, the cell viability is actually much lower, so they kill the cancer cell better way, and as compared to the non-covalent case where foliates are not linked conjugated manner. There are control experiments where there is no targeting unit present, so you cannot kill the cancer cell that effectively, or you don't at all take any doxorubicin into the mix. So this is very important, and presence of this kind of DNA in the corona area has added effect, because DNA normally has high persistence length, and that actually increases the surface accessibility of such kind of foliate receptor, targeting cancer cells. Another thing, most important thing about using this kind of DNA block copolymer for drug delivery is that here the DNA can be formed very easily with the DNA synthesizer, and that means it actually circumvent the multi-step synthetic challenge if in case you are using some other kind of moiety for this micelle formation. One can also form DNA multi-layer film or hollow capsule by using layer by layer self-assembly pattern. So in this case, as you see from the cartoon, one can deposit positively charged polyethylene imine polymer onto the silica surface charge, and you can make it positively charged, and then one can put negatively charged polythymidine residue with 30 mar, then that get electrostatically adsorbed to this polyethylene imine moieties, and then can use a 2 block oligomer system like poly AG or TC. Now they are complementary to each other. So now what can happen is that poly T30 that can actually form hydrogen bonding with the poly A20, and then the rest of the G20 of the poly AG, that can act as a sticky end, and it can actually also bind to the next level. That's where the poly TC is there. So this is how you can actually grow layer by layer self assembly. And you can, if you look at the AFM image, you can see, if you, the left one shows the AFM image where you can see the film, add urea into it. So then what happens is that urea is added to disrupt this hydrogen bonding, and there is a remarkable difference in terms of the film morphology. Now when you put this kind of layer by layer self assembly on silica particle, and then you dissolve the silica particle using hydrofluoric acid or ammonium hydrofluoric acid and fluoride, then you can actually create hollow capsule which can be used as a nano container. So this inorganic nanostructure like gold nanoparticle, the silver nanoparticle, or mesoporous silica nanoparticle, so this all can be used for drug delivery application. But this also gold nanoparticle, as I said, that this can be used for drug delivery, sensing application, and geometry of this all these nanoparticle can be nicely tuned. However, many of these inorganic nanomaterial, as I said, is toxic, and they can actually accumulate in our body. So and also it is very difficult to be degraded. Now also another very important thing is that this nanoparticles, inorganic nanoparticle, is it's very difficult to have in 2-dimension or in a very template ordering ordered pattern. So for that there is a great need of making hybrid material where you can actually use this organic material as a scaffold for this inorganic material, and they can actually somehow interlinked by some non-covalent interaction to form this kind of scaffold molecule. Just like using DNA or protein, you can actually make one-dimensional metal in a very long range order, or you can actually have if you put the organic scaffold as DNA or some polymer, you can, based on the interactions and specificity of interactions between the organic scaffold, you can also have these molecules or the inorganic nanomaterial spaced at a definite location, or it can be also spaced in 2-dimension. So this is very important, and we would now look for this kind of inorganic organic hybrid, so that you can actually get more control on increased dimension. So it has been known in literature that how to form a metal nanostructure. So you can take metal salt, and if you reduce the metal salt, you can form this colloidal suspension of those metal. Now they have a tendency to aggregate and then precipitate, but somehow if you can arrest the growth of those metal colloidal suspension by providing some electrostatics interaction or capping them with some ligand, then you can actually form metal nanostructures. And now depending on the type of seed of the metal kind of nanostructures, or depending on the ligand you employ to arrest the growth, you can actually form different kinds of shape of this kind of metal nanostructures. It's not only metal nanoparticle, you can have nanocube, nanocluster, nanorod, nanostar, and all these different, or you can also have core shell nanostructures. Now it's interesting, there are a lot of things known about these metal nanoparticles, and if you also change the ligand on the metal nanoparticle, and they can be used as therapeutics or sensing. So many of these metal nanostructures like gold nanoparticle or nanorod or silver nanoparticle, they have a unique property called surface plasmon resonance. This is a unique optical property demonstrated by the pool of electron in the metal. So if you have a spherical nanoparticle, you can see that you get very characteristic one surface plasmon band. But if you have a rod-like structure, you can see those electron, they can actually oscillate in 2 different directions. It can be longitudinal or in transverse directions. and based on that you can see there are two different types of band in surface plasmon resonance which comes. So this is what you can see on the right hand structure. Based on the shape and the size of the nanostructure, you can actually tune this wavelength of the surface plasmon resonance, and that is what is very much visible in the color. So you can have very different colors if you have different shape and size of the nanoparticle. And in fact, this is interesting because if you look at another example where you have a free gold nanoparticle, it gives a surface plasmon resonance like red color. But if you put a particular ligand which actually can bind to a particular surface, then upon binding it can give a different surface plasmon resonance. So this can be used as a sensing or therapeutics application. So it has been known in literature how to form a metal nanostructure. So you can take metal salt, and if you reduce the metal salt, you can form this colloidal suspension of those metal. Now they have a tendency to aggregate and then precipitate, but somehow if you can arrest the growth of those metal colloidal suspension by providing some electrostatics interaction or capping them with some ligand, then you can actually form metal nanostructures. And now, depending on the type of seed of the metal kind of nanostructures, or depending on the ligand you employ to arrest the growth, you can actually form different kinds of shape of this kind of metal nanostructures. It's not only metal nanoparticle, you can have nanocube, nanocluster, nanorod, nanostar, and all these different, or you can also have core shell nanostructures. Now it's interesting, there are a lot of things known about these metal nanoparticles, and if you also change the ligand on the metal nanoparticle, and they can be used as therapeutics or sensing. So many of these metal nanostructures like gold nanoparticle or nanorod or silver nanoparticle, they have a unique property called surface plasmon resonance. This is a unique optical property demonstrated by the pool of electron in the metal. So if you have a spherical nanoparticle, you can see that you get very characteristic one surface plasmon band. But if you have a rod-like structure, you can see those electron, they can actually oscillate in two different directions. It can be longitudinal or in transverse directions, and based on that you can see there are two different types of band in surface plasmon resonance which comes. So this is what you can see on the right hand structure. Based on the shape and the size of the nanostructure, you can actually tune this wavelength of the surface plasmon resonance, and that is what is very much visible in the color. So you can have very different colors if you have different shape and size of the nanoparticle. And in fact, this is interesting because if you look at another example where you have a free gold nanoparticle, it gives a surface plasmon resonance like red color. But if you put a particular ligand which actually can bind to a particular surface, then upon binding it can give a different surface plasmon resonance. So this can be used as a sensing or therapeutics application. Nanostructures, there are carbonaceous nanostructure. It can form carbon nanotube or fullerene, or it can also form graphene. Now these all employ carbon in the sp2 hybridization form, and because of this electron in the p orbital, these materials, they have very high thermal conductivity, and the carbon nanotube is known as one of the most stiffest material, better than steel, if you also take into account the lightweight of the nanotube. And these nanotubes, if you can orient themselves in a particular direction, and they can produce electrical property, and thus it can be also used in different applications like sensing. Not only this hybrid material offers increased dimensionality to the new hybrid material, it also can offer a plethora of applications which can include something like changing the surface plasmon resonance of the nanomaterial upon loading this organic scaffold, and then that becomes very sensitive. Something like if you have ligand receptor interactions or DNA hybridization, antibody characterizations or epitope mapping, or identification of some particular disease biomarker, so that can also change this surface plasmon resonance. So various things can actually change this surface plasmon resonance, and you can also detect single molecule using these nanomaterial and organic like DNA as a organic scaffold. Well, it can also like you can also use it for protein or nucleic acid biosensor. So other than that, it can be also used for like improve magnetic resonance property. Like you can also have you know MRI contrasting agent, which can be actually higher generation of MRI contrasting agent you can have if you are using this hybrid material. You can also have the different fluorescence property, very sensitive imaging, which actually comes out of this hybrid material. So we would now look at more in details how you can actually form in different scaffold using different kind of organic scaffold. You can assemble these nanoparticles, and then you can have increased properties so that you can actually use for some sensing application or um, drug delivery. So now we see some of the interesting hybrid material. Now this is a hybrid material coming out of DNA and the gold nanoparticle, which eventually can form 2D network like structure. And here in this case you can see that you can take a smaller gold nanoparticle, which is 8 nanometer, which has a particular DNA strand attached to it, and in the second gold nanoparticle, which is 31 nanometer, which is much bigger one, and it is color coded red. So you have the red color coded DNA strand 2, which is acting as a ligand in this case. If you take a template, which is a linking DNA strand, which is strand number 3, which has both complementary DNA strand of 1 and 2 nanoparticle, this thing, a ligand, then what can happen, it can actually bridge these two different nanoparticle, and that's what you can bridge them. You can see the UV spectra, UVs, the surface plasmon resonance. Dispersed nanoparticle has higher absorbance, but once you assemble the nanoparticle, it goes down, and you can see, since this is because of the hybridization of the complementary DNA strand, if you heat it up now at that melting temperature, DNA actually, the hybridized DNA structure then again gets separated into single strand, and it's very sharp transition. Now the TM image shows when you link this big and small nanoparticle, it can form very nice satellite structure, where the bigger nanoparticle is surrounded by the smaller gold nanoparticle. In the control case, when you don't add oligonucleotide 3, where you have the complementary strand, you add any other oligonucleotide, you can see this big nanoparticle and smaller nanoparticle, they don't get joined together, and then they stay as a separate assembly. Another example is patterning gold nanoparticle with a controlled spacing distance between these gold nanoparticle using DNA gold nanoparticle hybrid. So here you can see that there are two different DNA strands, and one can ligate one gold nanoparticle to this DNA strand, two different DNA strand. And then what happens, if you take a long-term DNA template, this DNA strand can actually go and bind to the long DNA template, and depending on the complementary sequence, and choose the long template such a way that you can actually position these gold nanoparticle. The hybridization will happen in different segment of the OTN, and you can actually position different dimension. In the first case, you see that you can have a head-to-head assembly, which has a 6.3 nanometer, or you can also form something like trimer, like 3 gold nanoparticle can get assembled onto the double-stranded DNA. So this is a very nice example where you can have very controlled distance between the patterned gold nanoparticle. So there's a nice example where you can rationally assemble gold nanoparticle. So in this example, the gold nanoparticle of 30 nanometer size was taken and it was modified with a specific ligand, which is a oligonucleotide. Here there are two different oligonucleotides were taken and that was modified, which are non-complementary to each other. Then they were combined. These two different nanoparticles were combined. Now they show very nice surface plasmon resonance. Now when another DNA duplex with the sticky end, which is complementary to the each of the ligand, were added, then it actually assemble all the DNA nanoparticle, and through hybridizing those sticky end and the complementary strand, and it can form assemble structure, and what we see is that the entire assembly precipitates, and the surface plasmon resonance from red color, it becomes bluish color, and that eventually precipitates. Now this aggregate, it was cycled with heating and cooling. You can see a very nice behavior when this aggregate DNA linked gold colloid, they were heated up to 80 degree, and you can see that if you monitor at 260 degree nanometer, you see the absorbance increases, and this has to do with this the base pair contribution from the nucleobases. And if you also look at 700 nanometer, which corresponds to the surface plasmon resonance of the assembled nanoparticle, now that gets broken, and then the absorbance goes down at that 700 nanometer. But again, if you cool it down, then DNA gets hybridized, and you see that 260 nanometer absorbance decreases. However, it again forms this pinkish blue color, and then you see that AUNP aggregate. Now this TEM image shows very nice assembled network of gold colloid which are separated. If you magnify the image, you can see that they are separated by 6 nanometer distance. So the unique property of this SPR, surface plasmon resonance, to change while assembling nanoparticle can actually be useful in sensing different biomolecules. So here you can see when these nanoparticles, they are staying independently, it shows red SPR, but as soon as they come together, they show purple SPR. And this is also visualized by the melting study. You can see that as you go higher and higher temperature, melt the DNA hybridized structure, and you can eventually go to red SPR because the nanoparticle becomes independent. But at the lower temperature, their nanoparticle, they show bluish purple like structure. So this is where the interparticle distance, they actually control the color of the DNA gold nanoparticle aggregate. So this is a classic example where in different cases, different kinds of oligonucleotides have been taken. The first case where there is a complementary target with the DNA oligonucleotide complementary base, you can see that they form more or less a purple like SPR, but with increasing temperature they can form red SPR. But if you have no targeting, then you can see that it never forms this bluish SPR. That's what you can actually check by, if you are half complementary or two pair base mismatch, how much this shifting, this changing the color or the shifting the SPR wavelength holds. And based on that, you can actually detect and differentiate oligonucleotides with very specific sequence, and this can be used as a strategy, can be used in a combinatorial approach for more specific, sensitive and reversible detection of oligonucleotides. This slide shows a classic example of DNA templated assembly to form conducting silver wire. So in this case, two gold electrodes were taken, and then the distance between these two gold electrodes are around 12 to 16 micrometer. Now this gold electrode, when they were surface functionalized with thiol modified oligonucleotide, two different oligonucleotide was put in two different gold electrode, and but there is a long oligonucleotide with both the complementary sequence were added to it, then it formed hybridized structure. And eventually where silver ion was added, it actually got stuck to the phosphate backbone of DNA, and when it was reduced, it can form metallic silver deposit onto it. So the AFM image shows very nice silver wire which connects the gold electrodes, and this particular electrode, when they are connected by the metallic silver, it shows very nice characteristic ohmic behavior on electrical measurement. So students, let's summarize what we have learned from this module. So this was a module where we focused on mostly these hybrid kind of structure. So we looked at the hybrid of polymer and DNA. So how it can form very specific sized micelles, and how you can do structural engineering to put a complementary long-stranded DNA, and that actually can form spherical micelle to long rod-like micelle. So we have looked at the structural maneuvering of uh, you know let's say these hybrid structures, and next we also looked at some very basic and classic nanomaterials like metal nanoparticles, because they are very important, because they possess important properties like surface plasmon resonance. We understood little bit in details, and then from then on we tried to look at inorganic organic hybrid, so that we can actually use this DNA as a scaffold material in the as an organic part for this inorganic nanomaterial, and then how you can actually tune the surface plasmon resonance property, and how this particular property can be used for sensing application, like it can be used, as we have seen, that oligonucleotide detection, that is that Shad-Mirkin example. Next we also looked at how this DNA and DNA scaffold, complementary DNA scaffold, can be used to connect two different electrodes, and we can also have biomineralization based on, or templation effect based on the DNA scaffold, and which eventually can be used for DNA nanoelectronics or device fabrication. So this definitely, there are a lot of application then I could actually put in here in this in limited time. So there are number of things you can do. So I guess this more applied module would be really useful for you, and you can actually look at in different papers different examples of different hybrid material, and which actually gives much better property than the native nanoparticle or only the DNA nanostructure can give. Thank you.